Imagine if we could edit diseases out of our DNA. Imagine a pair of molecular scissors so precise that we can cut out genetic defects, insert beneficial genes, or completely rewrite the instructions inside living cells. That tool already exists. It's called CRISPR. In this video, we'll explore how CRISPR works and what it means for medicine, agriculture, and for how we think about life itself. I am Frederick Christensen, and you are watching Geek and Nerdy. CRISPR is a system adapted from bacteria that allows scientists to cut DNA at specific sites, remove or alter segments, and sometimes insert new genetic material. It gives us the power to edit life, but also raises deep questions. Let's zoom in. Picture a long DNA double helix, like a spiral staircase, tightly packed inside the nucleus of a cell. When the cell needs instructions to make a protein, an enzyme opens a small section of the DNA and copies one of its strands into a single strand of RNA, a process called transcription. Scientists have learned to use this same principle, the way RNA can match specific DNA sequences, in a powerful new way. Using an RNA guide molecule that matches, by base pairing, a specific DNA target sequence, they can direct a protein called Cas9, the molecular scissors, to cut the DNA at an exact spot. In other words, scientists can now, in some cases, edit the original genetic recipe for building proteins to make it more favorable. For example, if a genetic error increases the risk of cancer, that faulty section of the DNA could, in theory, be removed or corrected, preventing the error from being passed on or expressed in future cells. Now you may wonder, if that's the case, why haven't scientists just edited cancer out of our DNA for good? This is because cancer isn't caused by a single DNA error. Cancer isn't one disease. It's hundreds of different diseases with different genetic causes. But that's for another video. Moving on, Cas9 is only one version. There are also newer CRISPR-associated enzymes, Cas12, Cas13, base editors, that allow edits of single DNA letters without cutting the strand. But Cas9 remains the workhorse and the clearest illustration of how CRISPR editing works. So, where can we use CRISPR? Researchers are testing CRISPR to treat genetic disorders like sickle cell disease, beta thalassemia, muscular dystrophy, and inherited blindness. In some clinical trials, CRISPR is used ex vivo. Cells are edited outside the body and then reintroduced in blood or immune cells. Someday, in vivo editing inside the body may target tissues like liver, muscle, brain. CRISPR can be used to insert resistance to pests, disease, or abiotic stress into plants. It can accelerate the development of crops with enhanced nutrition, faster growth, or fewer chemical inputs, meaning less pesticides. Because CRISPR can make small, precise edits without introducing foreign genes, in some jurisdictions these edits might be regulated differently from traditional genetically modified organisms, GMOs. These powerful possibilities come with equally powerful challenges, both scientifically, ethically, and socially. Sometimes CRISPR cuts at unintended DNA sites, off-targets, which could cause harmful mutations. Even in correct targeting, the repair process might produce unexpected insertions, deletions, or chromosomal rearrangements. But changes can also be made to sperm, eggs, or embryos, making changes to future generations. That raises additional risk and moral accountability. Editing embryos to correct a disease is one thing. Editing for enhancements such as intelligence or appearance is quite another. Critics warn of eugenics, inequality if only the wealthy can afford enhancements, unintended societal pressures, and disruption of human diversity. Who gets to choose which genes are good or bad? Could gene editing worsen inequality, creating a genetic rich class? Informed consent is tricky. For embryos or future generations, they cannot consent. Global governance. Different countries have different regulations or moral norms. The playing God objection. Is it hubristic for humans to tinker with life at its root? Many ethicists argue for cautionary, careful frameworks and strong regulation. The World Health Organization, WHO, has a program on human genome editing to examine ethical, social, and legal challenges. The WHO published a position paper and governance framework on human genome editing. Scholarly debates weigh the potential to prevent suffering versus the risks of misuse, intergenerational justice, and long-term safety. CRISPR is still young. We are in the early chapters of its story. The potential is enormous. The power to erase genetic disease, boost food security, heal previously untreatable conditions, reimagine biology. But with that power comes responsibility. We must proceed with caution, rigorous safety testing, transparent regulation, public engagement, global cooperation. We should weigh not only what can be done, but also what should not be done. The question is not just can we edit life, but how do we edit life wisely? If humanity charts this path well, CRISPR could open a new era in medicine and biology. But if we are reckless, we risk unintended harm, inequality, or irreversible mistakes. The scissors are in our hands. Let's use them carefully.